Welcome back. Physics builds models. Chemistry builds structures. Biology builds evolutions. 3 ladders, 1 way of thinking. And today, we drop that thinking straight onto a real exam idea. 2018 VCE Biology NHT Regulation of Biochemical Pathways, Question 11. Today's focus: enzyme activity under different pH conditions, neutral, alkaline, acidic, and how structure, denaturation, and controls shape real experimental data. A group of students wanted to investigate the activity of an enzyme that catalyzes the breakdown of hydrogen peroxide into water and oxygen. The students measured oxygen concentration using an oxygen sensor. The oxygen sensor fits into the top of a conical flask, as shown in the photograph below. The original table compares 3 distinct experimental groups, Flask 1, Flask 2, and Flask 3, to test how different pH environments affect the enzyme's ability to catalyze the breakdown of hydrogen peroxide. By converting this into a variable analysis, we see that all flasks contain the exact same type, volume, and concentration of both substrate and enzyme. This confirms that the substrate and enzyme conditions are controlled variables. However, the pH level changes between flasks. Flask 1 is neutral, Flask 2 is high, and Flask 3 is low, identifying pH as the independent variable. The buffer solutions and the distilled water did not react with the hydrogen peroxide. All 3 conical flasks were at room temperature. The students recorded the concentration of the oxygen over a 5-minute period. A. State a hypothesis that the students could be testing with this experiment. 1 mark. Analysis and official answer breakdown. A good scientific hypothesis for VCE biology must clearly state a testable relationship between the independent variable 4 and the dependent variable dV. Independent variable: for the pH of the solution, neutral distilled water versus high pH buffer versus low pH buffer. Dependent variable, dV: the rate of oxygen production, a rate of enzyme activity. Key takeaway for students: ensure both the 4 pH level and the dV rate of O2 production for enzyme activity are explicitly included. Predictions can be directional, such as higher pH increases rate, or non-directional, such as changing pH alters rate, as long as the relationship is clear and testable. Acceptable responses, examples. General: a directional hypothesis. If the pH of the solution is changed, then the rate of the production of oxygen is altered. Specific: a directional hypotheses. Higher pHs increase the rate of production of oxygen. OR: a low pH does not result in the production of oxygen. Explain the differences in the results that the students obtained for the 3 different flasks. 3 marks. Key exam tips for students. Address all 3 conditions. In 3 mark, explain the differences. Questions where 3 setups are presented. Allocation is almost always 1 mark per setup. Skipping 1 flask guarantees losing a mark. Link graph trends to biology. Simply saying Flask 1 goes up higher than Flask 2 is data description, not an explanation. Always link data back to enzyme shape or function per denaturation. Marking scheme and structural breakdown. To earn all 3 marks, students must address the results for each of the 3 flasks by connecting the visual trend on the graph to the underlying biological mechanism, enzyme activity and denaturation. Flask 1, neutral pH achieves the fastest rate of reaction and highest O2 production because neutral pH provides the optimal condition for the enzyme to operate efficiently. Flask 2, high pH results in a slower reaction rate than Flask 1 because the alkaline environment alters the active site or denatures some enzyme molecules, reducing catalytic activity. Flask 3, low pH shows no increase in O2 production, flat line, because the acidic environment denatures most or all of the enzyme molecules, preventing the reaction from occurring. If someone challenges your conclusion by claiming that hydrogen peroxide decomposes spontaneously at room temperature, you can refute this by using a negative control. In this control setup, the enzyme is omitted, and 52 milliliters of distilled water is added instead of 50 milliliters to compensate for the missing 2 milliliters of enzyme solution. Keeping the total volume, 102 milliliters, and concentrations exactly the same ensures a fair test, proving that any significant oxygen production in the experimental flasks is due to the enzyme, not spontaneous breakdown. Model exam answer. Flask 4 acts as a negative control group to determine the baseline percentage of oxygen produced by any spontaneous breakdown of hydrogen peroxide without the enzyme present. This provides a direct comparison for all other results, proving that the increased rate of oxygen production in flasks 1 to 3 is catalyzed by the enzyme rather than occurring spontaneously. Draw on the graph below the results that would be expected for the production of oxygen in Flask 4. 1 mark. Why should the line look like Flask 3? Since Flask 4 contains no enzyme, no catalyzed breakdown of H2O2 takes place over the short 5-minute period. The initial almost equal to 21% reading reflects standard atmospheric oxygen present inside the sealed conical flask at the start. Without enzyme catalysis, oxygen levels remain unchanged, producing a flat, constant baseline identical to Flask 3, where the enzyme was completely denatured. Official marking breakdown, and how to draw it. To secure the 1 mark on the exam, students must draw a line that meets these specific visual criteria. Position and shape. A straight horizontal line, at approximately 21% along the entire 5-minute duration, overlapping, or virtually identical to the line for Flask 3. Starting point, must originate at the initial atmospheric oxygen level, almost equal to 21%, at time 0 minutes. Strict boundary, the line must not drop below the initial baseline percentage of oxygen. Suggest one further improvement that could be made to the student's experimental design. Justify your suggestion. 1 mark. Official marking breakdown and acceptable options. To gain the 1 mark, students must state one distinct improvement and link it directly to a valid justification why it improves the experiment. Here are key options accepted by VCAA, structured with their respective justifications. Replication or reliability. Suggestion: repeat the experiment multiple times, or use multiple flasks for each pH condition. Justification: to calculate an average and identify or reduce the impact of random errors or anomalous results. Testing a broader range of pH levels. Suggestion: test a wider, more gradual range of pH values, such as pH 3, 5, 7, 9, 11. Justification: to determine the precise optimal pH for the enzyme rather than just testing broad high versus low conditions. Quantitative pH measurement. Suggestion: use a pH meter to measure the exact pH value in each flask. Justification: to quantify the exact pH level of the solutions rather than relying on qualitative descriptors like high or low. Extending time duration. Suggestion: run the experiment for a longer duration, such as 10 to 15 minutes. Justification: to see if oxygen production plateaus when the substrate is completely depleted. Concise model answer: choose one for students. Improvement. Repeat the experiment multiple times at each pH level. Justification. This allows for the calculation of an average, reducing the impact of random errors and increasing the reliability of the results. Consider any experiment. Explain the difference between the accuracy and precision of measurements. 2 marks. Marking breakdown, 2 marks. To secure both marks, a response must clearly distinguish between the 2 key scientific terms using precise VCE terminology. Accuracy, 1 mark refers to how close a measured value is to the true or accepted value of the quantity being measured. Precision, 1 mark refers to how close repeated measurements, under the same conditions, are to one another, indicating consistency or reproducibility regardless of whether they are correct. Concise model answer. Accuracy refers to how close an experimental measurement is to the true or accepted value. Precision refers to how close repeated measurements are to each other, consistency or closeness of replicate data. In this investigation, a group of students explored how pH influences the activity of an enzyme responsible for breaking hydrogen peroxide into water and oxygen, with oxygen levels monitored over 5 minutes. The results revealed a clear pattern. The neutral pH flask produced oxygen rapidly because the enzyme functioned at its optimal conditions. The high pH flask showed a slower rise, as the alkaline environment partially altered the enzyme's active site, and the low pH flask produced no oxygen at all due to complete To strengthen their design, the students proposed adding a 4th flask containing only hydrogen peroxide and water, creating a negative control that would confirm any oxygen detected in other flasks was due to enzyme activity rather than spontaneous breakdown. This control would produce a flat line identical to the low pH flask. Further improvements, such as repeating trials, testing a broader pH range, measuring pH precisely, or extending the duration, would enhance reliability and detail. Finally, the students distinguished accuracy as closeness to the true value, while precision refers to the consistency of repeated measurements. At the end of every lesson, I want to leave you with one idea: learning becomes powerful when you climb the logic ladder. Physics moves from situations to models to equations. Chemistry builds from structure to property to reaction. Biology grows from variation to adaptation to survival. Different subjects, but the same underlying logic. When you learn to decode systems, build ladders, and construct logic, you don't just solve problems, you understand how they fit together. If you'd like to keep climbing this ladder with me, you can find more lessons, breakdowns, and reconstructions on my channel. Thanks for learning with me today.